isolation and identification of potential antibiotic-producing bacteria isolated from Mount Isarog forest soil. Researchers Maiko M. Calendacion, Laika S. L. Enverga, and Giselle Atole from Partido State University. Presenter Maiko M. Calendacion.
Good day to our research advisor, Ms. Giselle D.C. Atole. To the Board of Judges, I am Michael Mendez Calendacion, and I would like you to greet you all a pleasant day. We will be presenting our research paper entitled Isolation and Identification of Potential Antibiotics Producing Bacteria Isolated from Mount Isarog Forest Soil. Introduction. Most novel antibiotics have been discovered by screening of wild isolates acquired from soil and other natural habitats. Soil microorganisms had always been the primary source for production of antibiotics and continues to maintain its significance. There are several soil microorganisms which can produce antibiotics, namely Bacillus, Actinomycetes, Streptomycetes, and Pseudomonas. Actinomycetes have been long recognized as primary producers of enzyme, antibiotics, anti-cancerous agents, and play a significant role in recycling of organic matter. Streptomyces are also known source of antibiotics for medical and veterinary and agricultural uses. Bacillus species are among the most investigated biocontrol agents, biopesticides which contribute to combat plant pathogens. Pseudomonas strains may have biocontrol activity, plant growth promoting activity, the ability to induce systemic plant defense responses, or the ability to act as pathogens. Objectives. Thus, in this study, soil microorganisms from the mountains were isolated, identified, and tested for antimicrobial activity. Specifically, it aims to isolate and purify bacteria from Mount Isarog forest soil, quantify antibacterial activity of bacterial isolates against selected pathogenic microorganisms, and to identify bacterial isolates with promising antibacterial activity. Framework. Soil sampling processing. Soil samples were collected from Mount Isarog forest soil. 10 grams of soil were transferred to 90 ml sterile distilled water and were stirred up. Different decay distributions from 10 to the negative 1 to 10 to the negative 4 was done. 0.1 ml of each dilution was spread-plated in nutrient agar plates that were incubated for 24 hours at room temperature. After incubation, colonies were counted and was expressed as number of colonies forming units or CF per gram of soil. After counting, separated colonies were strict-plated to nutrient agar plates and incubated for 24 hours at room temperature to obtain pure culture. For the antagonistic activity, agar wall diffusion assay was done using sterile and well-heated agar media. Test the organisms such as Bacillus subtilis, Escherichia coli, and Staphylococcus. Ciprofloxacin was used as positive control and sterile distilled water as negative control. All bacterial isolates were identified through DNA sequencing and analysis. Culture of the bacterial endophyte was sent to be with scientific solutions for identification. Results in discussion. For 3 sample sites were selected with 100 meters apart. The first site was surrounded by ferns and trees. The second site has abacus and grass vines, while site 3 was surrounded by tree, bamboo, and grass plants. For the plate count as a conduct, it was observed that each soil samples collected from the 3 sites has a diverse bacterial isolates based from the observed colony morphology. The results of the microbial numbers of each site has shown that site A has the highest observed CFU or ml with 2.35 times 10 to the negative 4, while site B had the lowest with 7.5 times 10 to the negative 3 CFU per ml. One of the factors that affect the CFU of microbial load is that the phylogenetic groups to which the isolated colony-forming bacteria belong. Growth of bacteria is affected by many environmental factors such as temperature, soil pH, soil moisture, and light. The other factors could contribute to the diversity of bacteria in the given space were as follows: growth, plant species, fertilization and irrigation, soil particle composition, land use, and management. Antagonistic activity. The diversity of these bacterial isolates found in soil could also mean a diverse activity of these isolates that could be useful in standard industrial and medical application. Because of these bacterial isolates from the three selected sites mentioned above were screened for the antagonistic activity. Using the agarwal diffusion assay, three test pathogenic microorganisms, namely E. coli, S. aureus, and B. subtilis, were used to evaluate the antimicrobial activity. Statistical data analysis. All data obtained was subjected to one-way analysis requirements and main differences were compared using the lowest standard deviation test as post-hoc analysis. P values less than or equal sign 0.05 was considered as significantly different. Statistical analysis of the data was done using SPSS, statistic versions 2.0. Identification. Analysis of the identified isolates showed the most of the isolates belong to the genera of Stenotrophomonas, Delphia, and Bacillus. Based from the results, isolate C2 identified as Stenotrophomonas multipelia and isolate C11 identified as Delphia lacostris. Delphia lacostris has shown to be a promising candidate for the production of antimicrobial compounds. Based from studies, Stenotrophomonas multipelia is commonly known for its importance in agricultural applications, specifically as plant growth promoting bacteria. It was reported by Hani et al. 2005 that Delphia lacostris also possess a nitrogen fixing ability capable of suppressing the growth of rice pathogens such as Xanthosomas, Oraceae waved, Oraceae, and R. solani. From figure 5, all isolates are capable of inhibiting the nest organisms. However, they showed a promising activity against base activities. However, based from statistical analysis, there is no significant difference in the activities of the isolates from site A capable of inhibiting E. coli, B. subtilis, and S. aureus. Isolates from site B also showed a promising activity against the test organism. Isolates B13 showed the highest activity against B. subtilis, significantly different compared to the other isolates, while isolates B24 showed a significant difference in its activity against S. aureus. On the other hand, all isolates showing antagonistic activity against E. coli were not considered statistically significant from each other. For the bacterial isolates in site C, isolate C21 showed the highest activity against E. coli, significantly different from the other isolates. While for the activity against B. subtilis, C11 showed a promising activity but not significantly different from the other isolates. For the activity against S. aureus, it has shown that isolate C21 and C26 show the highest antagonistic activity but not significantly different from the other isolates. Conclusion. Antibiotics are widely distributed in nature where they play an important role in regulating the microbial population of soil, water, sewage, and compost. Multi-drug resistance in bacteria has also raised a serious concern, highlighting the need to find more antibiotics. In this study, bacterial soil were isolated and identified from Mount Isa forest soil. It was tested for its ability to produce antimicrobial compounds capable of inhibiting pathogenic microorganisms such as E. coli, B. subtilis, and S. aureus. Results have shown that isolate C22 and C11, identified as Stenotrophomonas multipelia and Delphia lacostris respectively, showed a promising activity against this test organism. These results could further investigated to understand the scope of its activity and the type of compounds they can produce. Recommendation. Since the study had only focused on the isolation and identification of potential antibiotics producing bacteria, it is recommended to test defined chemical properties of soils such as temperature, pH, soil moisture, soil texture, and soil types before collecting soil samples. Furthermore, future researchers may also test the biochemical and characterization for further identifications of bacterial microorganisms. There are isolates tested that show no inhibition and some show partial inhibition. Thus, it is recommended for future researchers to test in other pathogenic fungi microorganisms to evaluate the effectivity of antibacterial activity. Furthermore, Future researchers may also check on the ability of the enzymes for enzymatic activity.
Thank you so much, Mr. Maiko, our presenter. We now proceed to the question and answer portion. Let us have Ma'am Marian.
Yes, thank you very much for that very interesting presentation. By the way, I'm also working with microorganisms from soil. So, na-appreciate ko ang iyong presentation. So, my first question is similar with our first presenter. Did you work on the same site where Malatunga was collected? Or this is different, totally different areas?
Different areas po, ma'am. Nag-ano po kami sa Barangay Pedro Arorogawa, Camarines Sur po.
Okay, but this is a different site. So another question is, did you secure permit for the collection of the soil sample?
Yes po, ma'am. May ano po kaming letter po na ano po signed by the barangay captain po ng nasabing barangay po.
So barangay level lang ang sa inyo. So hindi na pumapasok sa DNR regional. So ano lang, yun lang i-emphasize ko lang that since this is bioprospecting, we should follow the proper protocols in securing the permit since we are collecting samples from the environment and we are doing this for research. You've mentioned of diversity, no? I've heard that you said about diversity, that one side is diverse on this group of microorganisms compared to the other. In terms of saying diversity, have you conducted or calculated for diversity indices para masabi natin diverse talaga, no? Nag-calculate ba tayo ng indices?
Hindi po. Hindi po, ma'am.
I think it's better also that you compute or calculate for the indices para you can really say that once side is diverse with this group of microorganisms compared with the other sides. And then, how are you preserving? A similar question with the first kasi you were able to obtain potential bacterial stream. How are you preserving or maintaining these strains?
Bali po, ma'am, pinaserve lang po nung nakalagay lang po sa NA slants, then doon po po sa laboratory po, sa refrigerator po.
Oo, kasi baka sayang kung mawawala sila. So you can also deposit your microbial strains to other culture collections in the pantry, or you can do several other methods like literal stock or lyophilization. Hindi mababago yung activity ng inyong bacterial cells. Okay, congratulations and thank you. Back to our model.
Thank you so much, Ma'am Marian. Let's have Sir Edison.
Hi, Maiko. Thank you for your presentation. I am also interested dun sa study niyo. So it's nice to know that you have actually obtained some potential candidates for antimicrobials sa soil from it's a mountain area. Tama ba?
Yes po, sir.
So however, meron lang din akong very isa or dalawang tanong lang. Okay, so guys, Maiko, for the DNA sequencing, Ano ang sinend nyo sa Kinovet? Like, um, nagpakita kas ng figure kasi you have shown picture of a gel electrophoresis. Sarili nyong gel ba yon? Dun sa methodology part na notice ko yon. Is that your
No
own?
po.
Hindi. For ano lang, for presentation purposes. Okay, however, may tanong ako. What particular gene of interest na ginamit yung marker or pinasequence nyo sa Kinovet ba? Sa Kinovet. Anong gene ito for identification of the molecular identity of the organism?
Yung ano po, um,
So nag-PCR kayo,
yung
tama ba?
partial 16S rDNA.
Oo, partial 16S. Yung partial
Yes.
would be how long? How long yung partial? We all know that the full sequence of the 16S would be approximately 16,000 or 1,600. 1,600. Pero yung partial na, yes, tama ka, 16S siya. Pero how long yung pinasequence? Or do you have the name of the primer that you have used to amplify the gene of interest?
Nasa ano po, sir, nasa results po doon sa galing po sa UP po.
Galing sa
Naka-indicate po doon sa sinubmit na results. Galing po sa
Okay, so meaning, going back to my question, ang sinend niyo sa Kinovet would be the amplified or the amplicons or the PCR products, tama ba? Or the genomic DNA. So para hindi tayo masyadong nako-confuse. Yun yung tanong ko. Diba, sa process nyo, in your methodology, you have obtained your
no
sample.
po, sir, DNA extract lang.
DNA extract. Sila na ang nag-PCR. Sila Kinovet na. Tama? At nagbigay na lang sila sa inyo ng sequence. Tama ba, Aiko? Can you understand yung tanong? So, pagka kuha ng pure culture, extract ng DNA sa pure culture, so genomic DNA, igay agad sa Kinovet. Tama? Or wala, hindi kasi clear dun sa methodology kung ano yung sinend sa Kinovet. Like, if amplicons or PCR product or the genomic DNA. So, yun yung tinatanong ko. And related to that question is that, how did you arrive to the identity of the organism? Binigay ba sa inyo ng Kinovet yung identity o kayo ang nag-blast or nag-search ng matches sa GenBank? Nawala po ako. Miss Moderator, ako po ba yung nawalan ng connection?
You are in good connection. However, our presenter is not responding to the question.
Sige.
Yung connection.
So, yun yung tanong ko. Maybe pwedeng i-type na lang yung sagot kasi gusto ko lang malaman. And what's good about kung tama nga yung identity at na-ascertain natin na Delphia yung isa sa mga isolates ninyo, would you know that that particular species ay ginagamit for bioremediation purposes? So, aside from your antimicrobial result na maybe in the future, kung na-store nyo nga ng maayos, as per Ma'am Marian na tanong kanina, you may also use that isolate kung tama yung percent match natin kasi gusto ko talagang makita to further your studies. Ito naman, this time, bioremediation capacity ng isolate niya. Yun lang. Thank you. So, kung masasagot lang yung tanong ko kahit sa chat na lang. Thank you.
Okay. I hope you get that, Mr. Michael, our presenter. So, do we still have a question from our judge, Ma'am Kath?
Actually, more of because my question is also related to the previous judges. So I'm very much particular with the preservation technique that they are using for their isolates because yun nga sayang. So maybe they can tap nga my biotech any uh, microbial collection unit or ano po, any one, yun nga, sa biotech marami pong ganyan. So, also, yun nga, the primer that they used, also, yung gusto ko ring malaman, yun yung primer pair that they used. I think related with Mr. Edison question a while ago. Yun po, very much important also. So, medyo related po sa mga tanong din nila. Ayun po, I hope he can capture that also we can have the answers to the questions even kahit po sa chat. Thank you so much. But it's a very interesting, especially yung location. It's Mount Isarog, which is also close to my heart because I'm a native of Goa. So, congratulations in his presentation and kudos to the advisor, Ms. Lisel. Thank you.
Thank you so much to our third presenter. Now we proceed to the fourth